Hello. Hello. Am I audible?
Nope, you're fine. We can hear you.
Okay. I hope you can see my screen.
Yes, we
Yep.
can.
Okay. Hi, everyone. My name is Joy Banik. I'm proud to present my master's thesis research. I would like to thank my teammates for all the support and my guides, Dr. Elias Pedero and Professor Carides. I would also like to thank USGS Salinity Lab and Dr. Todd Shacks for making this research possible. This research has two parts, the initial data collection and the other is sensor-directed sampling. Let's talk about the data collection and the data fusion in modern precision agriculture. We will try to use the remote survey platforms, which are equipped with multiple sensors. In the next slide, I will show you the remote sensing platform to help your imagination. What we want to do is to have the ability to sample data at our desired resolution frequency. For that, I introduce you to short vectorization approach. Here you can see the L-band radiometer with like golden chips for the antenna. There's also gamma sensor near the foot of the passenger inside the ATV. And the ATV is also equipped with GPS sensor on the center top. Some of the problems that we face is that the multiple sensors are made from different brands and they don't always communicate well with each other. They have different operating frequencies. Some of the sensors take higher resolution data, and some of the sensors they sample data at a lower frequency. So the other problem is the internal clock, which do not account for the daylight savings and time dilation, etc. Sometimes they also have slight time mismatch because of the second we take to push the buttons to turn them on. Also, not all the sensors are collocated together, as you saw in the previous slide, that they are equipped at different positions in the ATV itself, and the GPS marks them at a different location. Some sensors also have a different uh, observation point offset, like they are equipped on a drone or a ATV and they read the data from a distance. So how do we come over all these problems? We create short vectors out of streaming data. While streaming, we can buffer at least 3 data points to create short vectors. And this allows us to remove any outliers on the go. And we can detect anomalies and disturbances in the sensors and remove them on the go. While we are able to remove the observation point offset, the direction is also important. That's why we consider vectors in place of just straight lines. And from those vectors, we create vector fields, which can also account for a Kriging uh, plane from where we can resample the data points at our desired frequency. We have the vector field and we can use ArcPy, which is a ArcGIS Python package. And my software is built on top of that. So for the ESAP is the sampling design, which is sampling software by Leash, and it's available on the USDA website. I'm also doing the sampling built on top of this ESAP software. So this is the points on a geospace for the sorry, yeah. This is the points. On the left side, you can see the variable space, the points, and the red points are the target points that we want to sample the data from to capture the variance of the data. On the right side, you can see the same data points, but on a geospace, which is the latitude and longitude. So we want the target points, we want some points that are nearest to the target points on the variance scale, variance space, but on the geospace, we want them as far as possible. For that, I created this small formula. Weight W is the weight, which is actually the weightage we want to put more on the maybe on the geospace or on the variance space. We can adjust that, which goes from 0 to 1. dk is the distance between the target point and the sampling point, and the dg is the distance between the points on a geospace. And with the help of this scoring formula, we can score all the points and select the point with the maximum score. This will help us to find sampling points which are not only spread out in the geospace, but also capture the variance in the sensor data to map to the ground truth. The future work would include making this into a Python package, Python library, which is built on ArcGIS, and also use an stochastic process to optimize this because this is the NP hard problem. It's difficult to calculate all the possible combinations. Thank you. If you have any
Excellent. Thank
questions,
you. Yes, we will be. We'll tell you time at the end for all the questions. This is a lightning talks, and so next up
Sorry.
will be uh, Rachael Cohen, and they will be discussing using GIS techniques to test a model of coexistence between two montane ground squirrels.
Hi, yeah, my name's Rachel. I was able to spend this past summer at the Rocky Mountain Biological Lab in Gothic, Colorado. Yeah, using GIS techniques to test a model of coexistence between the golden-mantled ground squirrel, shown on the left, and the least chipmunk, shown on the right. Just a little bit of background of the ecological theory for my project, since many of you are probably not familiar. Basically, the main effects of predators on prey in an ecosystem are the consumptive or lethal effects, the predator actually like eating the prey animal and removing it from the population. But there are also a wide variety of non-consumptive or non-lethal effects, all stemming back to the ecology of fear. Basically, the prey being scared that a predator may be around can impact their behavior, decision-making, habitat use, foraging, and a wide variety of other factors. So I looked at these non-consumptive effects in the least chipmunk and the gold-mantled ground squirrel in Gothic, Colorado. Previous research done by my mentor, Rosemary Smith, found that the least chipmunk is more efficient at foraging close to cover, as shown by the lower giving up density in this chart. A lower giving up density just means higher foraging efficiency. So because of their smaller size and their slower running speeds, they're a little bit more afraid of predators. They like to stay closer to safety, whereas the golden-mantled ground squirrel is a little bit bigger, but not quite as efficient of a forager. So they're able to do really well farther from cover because they're able to escape a predator more easily, but close to the edge of a meadow, the least chipmunk can still outcompete them. So what this means is that in a meadow where the maximum distance from the edge or max distance from cover is low, only the least chipmunk is present. And then in a larger meadow that has that area in the center that's more risky, both species will be able to coexist. And my mentor studied this by going out and measuring these meadows by hand with like a tape measure. So I obviously was thinking I could do a lot deeper analysis and just do it a lot easier with GIS. And that is what I worked on this summer. So I used two data sets created by Dr. Ian Breckheimer at the Rocky Mountain Biological Lab. The first was this basic land cover map, which I filtered to only include areas considered to be a meadow. And then the second was this canopy height map, which I filtered to only show vegetation less than 1 meter in height, because neither of these squirrel species like the vegetation to be too tall. And from there, I was able to identify all of these potential meadow areas where one or both species would be present. From there, I identified 34 meadows that were close to trails that I could go out with my binoculars and survey to see if I saw one or both species. And then using GIS, I determined the maximum distance from cover by applying repeated buffer operations at half-meter increments, so I could estimate to the nearest half meter the maximum distance from cover from the edge of each meadow. And so my results, of the 34 meadows I surveyed, 17 only had the least chipmunk, 16 had both, 1 had neither, and 0 had only the golden-mantled ground squirrel. This was pretty consistent with previous research. And using logistic regression, I found that the maximum distance from cover strongly predicted whether a meadow will have only least chipmunks or have both species present. And I was able to take this even further using GIS and look at the internal area of each meadow. So using the average maximum distance from cover for the chipmunk only meadows of 10.47 meters, I applied another buffer operation and used ArcGIS to super easily calculate that internal area. And I found that the internal area was significantly smaller for meadows containing only chipmunks compared to meadows that had both species. And to further illustrate this, here in pink is a meadow that only had chipmunks, and as you can see, there's only very small areas in the middle that are farther than 10.47 meters. That area is the area where theoretically a golden-mantled ground squirrel would be able to outcompete a chipmunk, but there's not really enough area to support them, so it makes sense that only the chipmunks were in this meadow, whereas in the yellow meadow, there's a much larger internal area, so it makes sense that both the chipmunks and the ground squirrels were able to coexist in this meadow. So in conclusion, GIS tools were super useful to re-examine this relationship between the golden-mantled ground squirrel and the least chipmunk. As a GIS minor at UCLA, I was really excited to bring my somewhat limited knowledge of GIS to this project. Not many other people at the lab really had any experience with GIS, so it was really cool to be able to try something that hadn't been done before. This kind of analysis, like I said, would not be practical to do by hand. To go out there I could not imagine going out there with a tape measure and trying to calculate like that internal area. It's so easy to just like click buffer in ArcGIS and do it that way. So this allows for really like quick and easy testing of new ideas and new questions with the data you have. Again, like I didn't have to go back out into the field and take more measurements. I could just click some buttons in ArcGIS and answer all my questions really quickly. And similar GIS techniques like this could be applied to a wide range of species and ecological systems. Thank you.
Thank you. That was very fascinating talk. Next up, we will not be recording the next one.
All right. Hey, everybody. Welcome. Thanks, Ryan, for that great introduction. As he said, I am the librarian for Geography, Maps, and Economics at UCLA. And this talk is going to be about lessons learned from a workshop I just taught called My Home Sank Quietly, Tuvalu Maps, Documentary Film, and Climate Change. So what actually was this workshop that has an awfully long title? This workshop opened with some introductory comments, and then we watched a 22-minute experimental documentary film called .tv. It's about the small South Pacific island nation of Tuvalu and how it is suffering due to climate change, particularly as it relates to rising sea levels. We then explored mapping that same nation of Tuvalu as it relates to climate change, and then opened the floor to a broader group discussion. Those are the 2 points we're really going to get into today. What was really interesting is that this had open registration, and my audience ended up being a group of film students, librarians, geography students, information science students, and history students. And these students were at all levels of education. We had undergrads, graduates, librarians who hold graduate degrees, as well as members of the general public who aren't currently students or UCLA affiliates. So that's a really interesting challenge when it comes to teaching GIS in a way that will stay interesting and useful to the folks in the room. One thing that was really successful was my quick and dirty intro to critical mapping before we actually started to explore maps of Tuvalu. We talked about how maps are not neutral carriers of information, how they're a form of data visualization, how maps contain intentional and unintentional silences, how they're dependent on the context they are placed in, and when reading a map to consider context, authority, bias, value, and perspective. I'm sure these things are not new for a lot of folks in this room, but it was a really good way to sort of set the stage right before we started looking at these maps of Tuvalu. Another success was taking the time to critically analyze this thematic map of Funafuti, Tuvalu, which is the capital of the Tuvaluan nation. Reflecting on those questions that I just posed on the last slide, we were able to talk about who is and is not represented in this map. We talked about potentially having unhoused people on the island who don't get mapped here due to not having a household to report. This is a map of hardship incidents by village and elevation by household for Funafuti Island, which, yes, quite the mouthful for a title. This is not a map that I made. It's a map that was created as part of a larger scientific study that I read in preparation for this. We also talked about why in this map some households are unsurveyed. There are dots for unsurveyed household, and there's no explanation in the paper for why they were unsurveyed. So why is that? That's something to look into as well. And we also talked about the perspective of this map. Was this map made with a Tuvaluan perspective by Tuvaluans, or is this non-Tuvaluan scientists looking in a world more unfamiliar to them? We talked about how potentially some Tuvaluans labeled on this map as living in a higher hardship incidence area. Would they consider themselves as facing significant hardship, or is this a label that was placed on them by outsider scientists? So we got a good discussion out of that. However, the biggest struggle came at the end section of this workshop that I led, which was the group discussion questions. The questions I had created ended up being too broad and not quite specific enough to get the group really going. It was really hard to try and wrap all components of the session into concise questions, which is part of why my questions were so broad, trying to incorporate the film and mapping and climate change. I was asking questions along the lines of what does it mean to map a nation whose borders are constantly shifting and shrinking due to climate change? How can we use maps to support climate justice efforts? And how does the medium of information impact how we think about climate change? There was a shyness also from the undergraduate students in the room in particular, but not exclusively the undergraduates, in giving responses to these questions about really big, broad ideas. The lessons learned and moving forward, the film was a hit. I have kind of an oddball taste in movies. Don't be afraid to get weird with it. I was really psyched that everyone ended up getting into it. I'm going to keep providing critical mapping introductions and applying it to relevant examples of mapping for workshops like this in the future. And I plan to ask more specific and focused questions during the discussion and then allow the responses to broaden the discussion and have the class take things in directions that they're interested in, as opposed to me trying to start broad, thinking that my peers in the classroom will take part of the broadness of the question and run the discussion from there. So that was the sort of opposite approach that I took, but it's something I'm going to try for this type of teaching in the future. So thank you all. I'll drop this link in the chat to a list of readings that I used for this discussion on Tuvalu. They're really interesting. And that's my email if you want to contact me. Thanks, y'all.
All right. Thank you, Maggie. Next, I don't know. I've not seen him in the chat. Alexander Brown. If you are here, raise your hand. Nope. All right. So we will go on to the next lightning talk. Tianrun Li, who will be discussing spatial variability and plant heat damage within South Fork American River National Park during summer.
Hello, everyone. Can you guys hear me clearly?
Yep, we can hear you, and we can see your little slides as well.
Okay, cool. So, yeah. Let's start it. Hello, everyone. My name is Tianrun Li, and I'm now a third-year undergrad visiting student at UC Berkeley. And today, I'm gonna talk about the spatial heterogeneity in plant heat damage within the South Fork American River area during summer. So let's have a look at the background first. Accumulating evidence around the world have highlighted increased mortality risks for trees during severe droughts, particularly under warmer temperatures. And California, South Fork American River area in specific is often threatened by heat waves along with droughts and wildfire. Consequently, cohesive approaches have been taken to tackle with the plant heat damage in this area. But it's also of great importance to have a closer look at which part of this study area suffered most in terms of heat damage. This is a rough land classification, and those in darker green shows a higher vegetation density, while those area in light green has little vegetation density. And those area in yellow are mainly background clustered around the eastern part of the studied area. In this study, I was able to showcase the NDVI difference between August and April. I downloaded multiple Landsat images from USGS and made a deduction between the NDVI value in August and April for 2023 and 2013. These two images has similar patterns. Those area in red shows that their vegetation have been damaged more severely by the heat wave, while those in yellow shows little to no change in NDVI difference. And astonishingly, the eastern part of this study area actually has a higher vegetation coverage, and the vegetation there are healthier in the 4-month span. We think the most important reason behind it is the elevation, which contributes to a cooler temperature and made temperature in summer more adaptable for them, more suitable for the vegetations there. However, if we compare those two images together, we can see that there are both vegetation loss in, the heat damage was more severe in both red area and green area, which is concerning. In the remainder of the time, I will introduce some research that is closely related to this topic, which was conducted by me in West Temu Natural Reserve in China. We collected, we've chosen 7 sites at approximately every 200-meter interval, and we collected 115 species samples and took some measurement on site in the field, like some tree heights and diameter at breast height, and bring those samples of branches and leaves back to laboratory for further measurements. We measured the leaf data and branch data. The image on the right shows the microscopic observation of stomatal density and size. By collecting those functional traits data, we can use principal components analysis under this LHS scheme, which is also known as Leaf Height Seed Plant Ecology Strategy Scheme. And this scheme was proven to be effective as a research conducted in southwestern USA. By doing so, we can distinguish which type of local species are more adaptable to a warmer climate, which can be used as a great information for vegetation recovery after heat damage. And that's all for today. Thank you so much for your attention.
Right, thank you very much. Very good presentation. And next we have Isabella Santorsiri, who is going to be talking about outwitting a nest parasite, suitability model development, and field verification for the brown-headed cowbird. Well, I guess she was here a minute ago. Maybe we have some technical problems. So we will skip ahead to the final lightning talk that I have on the list. One I was kind of looking forward to because it involves sharks. Jorge Garcia will be talking about shark attack data now analytics with R and Kaggle. It will hopefully not terrify all of us going into the water.
Oops. All right, can you see the screen?
Yep,
Yes.
we can.
All right, no terrifying stuff here. So this lightning talk demonstrates using the R programming language on the Kaggle data science platform for analyzing and visualizing records from the global shark attack file. So the agenda today, I just want to go over a few questions. Why study shark attacks? Why use Kaggle? And why use the R programming language? And finally, why use spatial data analysis? So sharks are amazing, intelligent creatures. They are not mindless human hunting machines. Some sharks allow and even seek out human interaction, and yet attacks do occur, hence the need for analyzing shark attack information. So why use Kaggle? Kaggle allows you to define extensive metadata about the data sets you upload. They allow you to explore your data without having to write any code initially. And you can also share your data with others as well as use data uploaded by other Kaggle users. Kaggle also supports notebooks. Notebooks allow for easy outlining and documentation of code. Other Kaggle users can copy and use your code, and notebooks are a great way of providing analysis and observations in line with your code and your visualizations. Kaggle comes with many, if not most, of the spatial packages already installed and available, both for Python and R. Reading spatial data from a variety of formats using R is quite simple. So R is typically used as a statistical language, but it's also a great language for data wrangling, data analysis, and data visualization. So R is a well-supported language on Kaggle, and with R, you can create very interesting graphics with very little code. So in this example, we create a data frame from the spatial data frame, calculate a new variable, create aggregates for the 3-line plots, and finally use the plot and lines functions to create the chart on this slide. From this chart, we can observe that though attacks have risen significantly since 1980, fatalities have not risen at a commensurate rate. With R, it is easy to read in spatial data, convert the data into spatial data frames, and visualize the data using libraries such as TMAP. So in this example, we aggregate the attack and fatality variables by country, merge with the country polygon data frame by country code, calculate the min and max years for our title, and finally use TMAP to generate the map. So from the map, we can see that the US has the highest attacks on record, while South Africa and Australia are second and third. With R, it is also easy to create non-spatial visualizations to supplement your spatial data analysis. So this gives us the ability to produce an elegant box plot, which shows this distribution of attacks per year by species. And so from this plot, we can see that whites, tigers, and bulls have the highest distribution of attacks per year. Other non-spatial visualizations include bar charts, stacked bar charts, pie charts, and density plots. All these can be easily created using highly configurable R charting libraries. And finally, with R, you can also answer questions about the data using elegant aggregation routines. For instance, how can I tell which species has the most attacks per country out of the top 10 species with the most attacks overall? So to answer this, we subset the shark data frame by top 10 species, get a count of attacks grouped by country and species, sort the results by the count in descending order, group once more by country only, and finally collect the first row for each country, which should contain the species with the most attacks for that country. To spatially visualize the aggregated data, we again merge with the country's data frame by country code and then use TMAP to create the map. We now have a beautiful map displaying which of the top 10 species have the most attacks per country. From the map, we can observe that white sharks have the most coverage, while tiger and bull sharks come in at second and third. And that's all for my talk. Thank you very much.
All right, thank you. And thank you for not making me terrified to go into the Pacific Ocean.
No reason.
Excellent. And I don't think the other presenters have shown up. So thank you, everyone, for a great round of lightning talks. Thank you for a wonderful 2023 UC GIS. It's been a lot of fun. Thank you again for the other committee members for helping me pull all the stuff together. It's been really fantastic. So thank you. Enjoy GISing and go forth and map.
Thank you for your organization.